It's my pleasure to serve as the moderator of Panel 1, which is translating early development science into interventions and policies. Our first speaker is Dr. Fernando Martinez, who's a Regents Professor and Director of the Asthma and Airway Disease Research Center at the University of Arizona in Tucson. And the rationale for having Dr. Martinez present is while almost everybody who presents on the topic, and we saw it actually in the last presentation, and there was a box that said G by E, gene by environment. We understand that that's how the biology works, but there are few examples in which we understand, in fact, how it works, and the work that Dr. Martinez done will, I think, help us understand that there are ways forward in both the science as well as using the science to translate into practice. Welcome.
Thank you very much, Pat. It's a real pleasure to be here, and I thank Pat for giving me the opportunity to present some of the work we have done in an area that is completely unrelated to the one you study, but you will see in a moment that it is not completely unrelated. My work is funded by the National Heart, Lung, and Blood Institute. When people think about environment and genetics, they usually think about them relatively independently. In other words, they think that when you study genes, you can study genes without really having to study the environment. They don't tell you that explicitly, but any GWAS that you may have seen usually does not make any evaluation of the environment. And any study of environmental factors usually considers the genetics like a potential effect modifier, but doesn't take it into account. And therefore, it's like if we had a genetic blueprint that really is going to determine which phenotypes you have, and there is some alteration of that blueprint by environmental factors or vice versa, but both act as independent of each other. Or if you think about it in what is called a normal reaction, and I just put some, I'm sorry, I'm very ignorant about what you do, but let's say you think that child maltreatment in the first 5 years of life can be related to depression and you want to study it, but at the same time you know that genetics has an influence. If you're going to study both separately without telling us, this is the way you're thinking about it. Here is the child maltreatment, here is your outcome, depression, and what you're really telling us is that the two are independent of each other. So for any level of child maltreatment, the genotype is going to have an effect, so I don't need to study child maltreatment to determine the genetics of depression. And I don't need to really study the genetics of depression to study child maltreatment, but because for any genotype, you're going to have exactly the same effect, or relatively the same effect. I exaggerated there, but they go in the same direction. So all GWASs avoid studying the environment, and anybody who wants to study child maltreatment until now that I have seen with very few exceptions, and it applies to asthma, by the way, does not study what happens with the genes. In other words, what we think is that the genotype determines the phenotype, and I could have put here environment, and therefore that I can map the phenotype to the genotype directly without studying other factors that determine it. And again, most GWASs of asthma, for example, which is my area of interest, ask people, do you have asthma, or has a doctor told you you have asthma, and then study the genes, and we have fantastic studies that I will show you in a moment. Now, I have to say very honestly that there have been some successes with this paradigm. And one of them is height. Here is a study. Height is very easy to measure accurately. Very different, I think, to depression. So here is a study with 253,000 individuals, which discovered 697 loci in, SNPs, single nucleotide polymorphisms in 423 loci. A very good friend of mine, Jonathan Pritchard, a fantastic geneticist from Stanford, has called these traits omnigene traits, because in the end, if you study not 200,000, but 200 million people, every single gene in the genome will have variants associated with these variants. He calls that omnigene or omnigenetics, not polygenetics. But really, this has been a big success. What it has discovered, as you can see, is that almost every gene is involved in the genome. And it has had a success, because in the end, if you want to do this, you want to do it because you want to predict. And if you want to predict height, here is the predicted national mean in Europe for height. And here is the observed national mean in Europe for height. And with some variation, you can see that what you see with your eyes is true genetically, which is people from Portugal, Spain, and Italy are not as tall as people from the Netherlands. Now, we didn't need to do all this because we have our eyes, but it's good that we confirmed it. Genes can predict what happens to people. I hope that is why we're doing this, and not only to determine what proportion of our ancestry comes from Galicia, as mine comes from. The problem is that you get a little more complicated than height, and everything falls apart. You just go to weight, and you can't predict by the genes. The same thing that I just showed you was done for weight. Fantastic, a beautiful, I didn't show it to you because Pat said, oh, it's too long, so I took it out. But it's fantastic, it's perfect. The genetics is, there are hundreds of thousands of genes. I just want to predict, if you are from the Netherlands, you are not, if your genes correspond to that, you are different from somebody from Spain. Look how it works, it doesn't work. No correlation, you cannot predict where you're from. But there's only one reason why that could happen. It is because there has to be some disconnection between the genes and the environment. Because if it worked like I show you for the norms of reaction, it doesn't matter. I mean, you should be able to determine one or the other without having to study both. Weight, you have to study both. So the relation between genotype and phenotype is not a straight line. We're not fully and totally programmed by our genes. The genes are not a program. And I know that goes against everything you hear every day. They're not, because there are all these other things that influence this long and winding road. And I don't have time to talk about them. Epigenetics, integration, phenotype, a matter, by the way, that Galileo Galilei decided to study once they told him that he couldn't say anymore that the sun, that we went around the sun and not vice versa, and plasticity, which is what I'm going to be talking about. And I'm going to be talking about, in the last 10 minutes, about the disease of my interest, which is a disease with enormous disparities, which is asthma. And the way in which we have thought about this disease until very recently, until we've had a tremendous change in the way we understand it, is that the disease was caused by the factors that determined its attacks or its expression. That what caused attacks of the disease or the disease to get worse was what caused it. And for 50 years, we strenuously studied everything that we thought caused the disease because it caused its attacks. And here you see them all. Pollution, anger, stress, pets, exercise, pollen, bugs in the home, chemical fumes. All these things are everyday questions to my patients in my clinic. And we try to avoid it by avoiding these exposures, for example, to allergens, with extremely unsuccessful efforts, failures. Why? Because we had a natural experiment, as Dr. Braveman has just told us is important to listen to. And this is the natural experiment. It is a part of Europe that, funded by the European community, these farmers can keep their lifestyle. They have 5, 6, 7 cows. They live on top of their cows, as you will see in a moment. And it was discovered by a former postdoc of mine who got the highest science prize in Germany for these studies, that these people lived in such conditions that they were exposed to factors that were strongly preventive of asthma. Because when you study these people who have Mercedes and therefore don't have a very low socioeconomic status, they have 1/10 of the asthma of people who live in the same rural communities but are not exposed the way I just showed to you. And this was published in The Lancet some years ago and completely changed the way we understood asthma. Because now asthma started being not a problem of what you're exposed to, but what you're not exposed to. In other words, asthma is most likely caused by lack of exposures to the factors that are related to living in those conditions that probably establish the susceptibility then to those factors that determine the attacks. And they published in the New England Journal of Medicine many years ago this, which is on the x-axis and the toxin exposure, which is representative of bacterial exposure in the homes, which is obvious why, you just saw it in that picture, and the prevalence of Wheeze in the community. And you can see that it is inversely related. The more you're exposed to endotoxin, the less asthma you have. When I saw this at the time, I was studying this gene, which is the receptor for endotoxin. So I called Erika Formuchos, who was, as I said before, a postdoc in my lab, and I said, why don't we combine both things? Why don't we study this gene, which is the receptor for the environmental factor that you just showed prevents asthma, apparently? Because perhaps there could be variations in the receptor that could show relation with the exposure. It's the exposure, the receptor, and the outcome. We had it all. And she sent one of her postdocs, and our first analysis, here is a surrogate of asthma on the IgE on the y-axis, and there was absolutely no relation between this polymorphism and asthma in that community. Oh, what a disappointment. But then, see, this is what happens with obesity, as I showed you before. But then we said, wait a minute, let's divide the population among those that are more exposed or less exposed. Let's do a norm of reaction. And this is what we found. What we found was quite strange at the time I saw it. But then this was replicated by another 4 scientists in Detroit, in different parts of the world. So it has been confirmed in Manchester, in England. What you can see here is the same normal reaction I showed to you before. Here on the x-axis is the level of exposure to endotoxin, and on the y-axis, another surrogate for asthma, which is allergic sensitization in this case. And you can see that there are 3 curves there for the 3 genotypes that I was studying in Tucson. But the only one that really shows a very strong effect, a very strong protective effect, is the yellow one, which is CC. Instead, the other 2 show some effect, but are quite flat. And what that determines is that at low levels of endotoxin, it is the CC which is the risk factor for asthma. And at high levels of endotoxin, the CC is protective. So if you put all of them together in the population, they cancel each other and you don't see it anymore. But it also tells you that not everybody is equally susceptible to the protective effects of endotoxin. Because for some people, endotoxin is bad. Right? Because if you see it the other way, endotoxin doesn't do anything to CT and TT. And in fact, it's so flat that it doesn't protect you, and therefore, if you get asthma, it's bad for you to be exposed to endotoxin. Later, there was a genome-wide association study done for asthma. And this is it. And immediately, just without even calling me, Erika said, well, here are some of the most important genes that are associated with asthma. How do they work among these people who live in these communities in rural Europe? And this one was done with 24,000 cases and 120,000 controls. And this here is the most replicated gene for asthma that we have found anywhere in, and I will add, in non-rural communities, because of course it's much more difficult to do a genome-wide association study in a rural community because you have to go each little town asking people questions and getting DNA. So all these studies are done in cities. So what was found was that there was a gene strongly associated with asthma, which has been replicated in every single study among urban populations. And so Erika took this gene. I don't have time to talk what it really does, but we can talk about it in the discussion. Genotyped it in the rural communities that you just saw, and guess what she found? Nothing. It's the only place where this was not replicated. So they said, let's do the same thing that you guys did with CD14 for endotoxin. And this is what they did. This is the gene divided on the upper part between people who are exposed to animal sheds above and people who are not exposed to animal sheds below. And the extraordinary thing is that this gene is protective among people who are exposed to barns when it is a risk factor among people who are not exposed to barns. So why is it then that people who are exposed to barns are protected against the development of asthma? Because they have a genetic variation which if you're not exposed to Barnes, it's bad for you, and if you're exposed to Barnes, it's good for you. Now, why would nature do this to us? And I think it's a very good reason for nature to do that to us. Because what nature is doing is creating within the community the potential for surviving in the most different environments. We're not programmed to survive in a single environment. We're the result of evolution. And therefore, to finish, this is the way it really works. It doesn't work that we are predisposed to be good or bad. I have seen, quote, studies of the genetics of accomplishment in school. And I can assure you, they have not taken into account all the possible populations in which that can be studied, because not all populations are equally accessible. Like I showed before, not all populations live in cities, and if you study only cities, the gene in 17q is strongly associated with asthma, but it's not if you study everybody that is exposed to every potential factor that human beings are exposed to. So I think if you really want to understand, you need to understand, as Dr. Braveman was telling us before, that this stuff is really complex. The way I see it now is that genes are above, environment is below. Yes, there is a type A and type B for genes that, as you can see in this slide that I stole from a great evolutionary biologist, Dr. Levontin. Yes, type A and type B tend to be more towards phenotypes A and B. But with the right environmental exposure, type B can even be phenotype F, given the right conditions. And the same thing is for the environment. There are people who live in very adverse environments, but with the right type of other environmental factors and genes, they can be very successful and be phenotype A if phenotype A is being successful. There's finally a final factor that, because we cannot study it very well, is not taken into account, which is that there's also our noisy development. I mean, I've treated identical twins with the same disease, cystic fibrosis, and they have very different outcomes. So noise also plays a significant role. If we use this kind of an approach, and we understand that you cannot study environment without in some way or the other considering susceptibility, I think we can really plan for providing the right therapeutic or preventive approach to the right individuals with the right susceptibility, resilience, or risk. Thank you very much.